Good morning. I would like to welcome everyone to today's MD Anderson Cancer Center Enjoy Science seminar. This is a series of lectures given by the MD Anderson faculty members and organized by the Office of the Chief Scientific Officer, Dr. Giulio Draetta. I'm Alessandra Ferraioli, and I have the pleasure of hosting this series with my colleague, Dr. George Kalin. The goal of Enjoy the Science initiative is to strengthen the connections between the scientific and medical community at the MD Anderson Cancer Center and our colleagues, our patients, and our friends around the world during this time of limited in-person, at the bench, and in the clinic activities. Today's seminar is being recorded and will be made available on the Enjoy Science website later this week. The speaker of today is Dr. Sendurai Mani. Dr. Mani is a professor in the Department of Translational Molecular Pathology at the University of Texas MD Anderson Cancer Center. He is also the co-director of the Metastasis Research Center and the co-director of the Center for Stem Cell and Developmental Biology, also at the MD Anderson Cancer Center in Houston. Dr. Mani is an authority in the field of cancer stem cells with a particular interest in the molecular mechanisms favoring the development of metastases. And today, he will be discussing for us the topic of metastases, novel strategies to stop tumor spread. Dr. Mani, you can present your lecture. Thank you.
Thank you, Alessandra, and also thanks to George for inviting me and giving me an opportunity to share what we do. It's particularly, it's a wonderful forum to share what we do with the rest of the world rather than rest of just MD Anderson, which is a great idea. Thank you for doing that, to both of you, and also to Giulio for supporting this seminar series. Hope, again, thank you for the nice introduction. Today I just wanted to kind of send my appreciation for Dr. Fiedler, who passed away last week. I'm sure you all know he's a legend, and he has done a seminal contribution to the metastasis field. And today's my lecture is dedicated to him. He's a wonderful man, and we miss him. So my lecture today is going to have these following 9 components where I will be discussing various topics, but as we go along, we'll highlight each one of them. First, we want to understand why do we care about studying metastasis. So this slide is from global kind of statistics and prediction. What it shows is that today we have approximately 18 million cancer survivors in the world, but which is expected to go pretty high by 2040, right? And this is a cancer-related death today in 2018, but you could see which is going to be really high by almost 16 million by 2040. So this slide shows, for example, if somebody has a primary tumor, just let's say breast cancer, the survival rate for these patients is really high, which means, due to various techniques and medicine, we are able to cure breast and prostate cancer very well. But if the same patient or if a patient with the metastatic disease, we don't have any treatment. As a result, most of the time these patients die because of metastasis. And you could see that almost independent of the cancer type, if a patient do have a metastasis, their survival rate is extremely low. The reason is we don't have a medicine to treat metastasis, and we are in the process of understanding the biology of metastasis. So my lecture will focus on our contribution or what we have done towards understanding the biology of cancer metastasis. So this slide shows that the Brian Ellenboss and Bill Hahn, when they were at Whitehead Institute with Robert Weinberg as a postdoc, they took normal human mammary epithelial cells and they introduced SP40 early region, catalytic subunit of telomerase, and oncogenic form of HRAS. So when they did, they were able to convert these cells into cancer cells by various criteria. But the interesting thing is when they put these cells into the mammary fat pad of a mouse, they developed a tumor, but predominantly it was non-metastatic tumor, suggesting that this transformation is not sufficient to make tumor metastatic. So this is a picture of them when they were the Weinberg lab. So there's two papers which shows that one is on a kidney epithelial cell, another one is on a human mammary epithelial cell. This is true on many other epithelial cells, that the transformation is not sufficient to make tumor normal cell to become metastatic cancer cells. Rather, that is sufficient to convert them to a cancer cells. So right around that time, so metastasis is driven by separate set of genes and pathways. That's the kind of conclusion from this story. And right around that time, I joined Bob Weinberg lab and then wanted to know the biology of metastasis, because that's something which was less understood at that time. This is 1999. And so if you look at a metastatic process, it's quite complex. You have a primary tumor which become highly angiogenic, then the tumor cells manage to get into the vasculature, and then once they're in the circulation, they travel, and then they exit the circulation at a distant site, and they create a micromet or remain a colony, small colony at a distant site, and eventually some of these micrometastases develop to become fully vascularized secondary tumor or metastasis. So it's a quite complex process. So it makes sense that transformation makes a normal cell to become a cancerous, but not necessarily complete the entire cascade. So again, so Bob Weinberg, he has an amazing style of letting people in his lab to explore whatever they like. And so I got an opportunity to venture into this problem, and then I have been focusing on this since then. So when I joined the Weinberg lab, there's another postdoc. Her name is Jing Yang, who is a professor at UCSD. She also joined with a similar interest to understand the biology of cancer metastasis. So what we kind of did together at that point, we kind of identified all the genes which are known to regulate metastasis by that time. So we introduced them to the cancer cell generated by Brian Ellenboss and Bill Hahn and asked the question, can we convert them to become metastatic? So remember, in all our experiments, what we are doing, we are introducing cells into the mouse mammary fat pad and asking them to develop metastasis in the lung. Unfortunately, none of the genes we explored at that time was able to promote spontaneous metastasis. But those genes which we were using predominantly identified by a method called experimental metastasis. So what is spontaneous metastasis and experimental metastasis? So if you look at the slide, if you introduce the tumor cells to the mammary fat pad, and then if you allow the tumor to complete to all the way to the lung, then that's called a spontaneous metastasis. And then on the other hand, if you introduce cells through the tail vein, you are directly seeding the tumor cells to the lung, and that's called experimental metastasis. So those genes predominantly identified being a player in experimental metastasis. So when we try to introduce those genes into this the transformed cells, and then we assessed for a spontaneous metastasis, they were not sufficient. So the question at that time is that, can we identify novel genes which could regulate metastatic process? So we were lucky that Dr. Fred Miller, he generated the syngeneic isogenic mouse cell line, where there are 4 of them. These cell lines have a differing metastatic potential. All 4 forms a primary tumor. Only 3 goes to lymph node, 2 goes to the blood, 2 can be detected in the lung, but only 1 can develop secondary metastasis. So we performed a microarray at the time. Then we looked for novel candidates. Fortunately, we found that this is another data to show that they proliferate both the cell model. So we used primarily 67 and 41 for the rest of the studies, which is listed here. One can form a primary tumor. Another one can complete the entire metastatic cascade. So when you look at these two cell models, both can proliferate equally in a petri dish. Both form the same number of soft agar colonies. Both form a similar number of acini if you put them in an acini assay. But the 41 is highly migratory and the 41 is highly invasive based on void and chamber assay. So now if you inject them to the mouse mammary fat pad, the 67NR forms again primarily just a primary tumor and no metastasis. But if you look at a 41, they form a primary tumor, also develop metastasis, so which is evident here. So when we screened these two genes for differential gene expression by microarray, we pulled out number of genes, but two of them stood out pretty at the top of the list. One is called a twist, another one is called FOXC2. That kind of suggested that these genes may play a very critical role. So we went ahead and asked what is the role of these genes, and that's when we discovered that the embryonic developmental program, EMT, may be playing a critical role in converting these non-metastatic cells to become metastatic. So I'll show you the data in the following slides. So here we used an shRNA for twist, and you could see the control mouse where again the tumors were introduced into the mouse mammary fat pad or mouse breast, and then we are looking at a lung, and you see the control mice, you have several nodule, but if you knock down twist using shRNA, the metastatic nodule is significantly reduced. On the other hand, even though we are changing their metastatic potential, the primary tumor growth is not affected. So which is in line with our observation that the metastasis is controlled by a different set of pathways or genes, and the transformation is not. So the most striking observation was when we overexpressed twist in a human mammary epithelial cells, they changed from this cuboidal looking morphology to more spindle-like morphology. So same thing was true for FOXC2 as well. Here is the control mouse where you see number of nodules, but when you knock down FOXC2 using shRNA, the number of nodules is significantly reduced. If you overexpress FOXC2 in an epithelial cell, they tend to become more mesenchymal. Same way, the primary tumor forming capacity is not changed. Once again, confirming that these genes regulate primarily metastasis, but not the primary tumor formation. So that kind of made us think, and there was quite a lot of papers at the time also suggesting that EMT is a critical player for metastasis. So the way when you just look at this problem, if you look at a normal mammary gland, you see the cells are tightly attached with one another through various cell adhesion molecules. When they get transformed to become a cancer, they still attached with one another through expression of various cell adhesion molecules. So what they need to do is they need to break this cell adhesion by breaking, they need to break the cell adhesion. Also, they need to break away from the matrix, and now these cells can be free to move, and they potentially develop metastasis at the distant site. So if this is true, if I take those cells which were transformed earlier by Brian Ellenboss and Bill Hahn, if I introduce these EMT genes, can I make them metastatic? So that's what the study we have done. So when we introduced FOXC2 in this case, for example, this is done by Brett Hollier when he was in my lab. So he took this human mammary epithelial cells, introduced FOXC2, and then he put them in a mouse mammary fat pad. There was no, while the control cells developed a primary tumor, but you could see there is no metastasis. These cells are labeled with luciferase, and you could see lung, liver, brain, and spleen. There is no metastasis in any one of these organs. But on the other hand, if you express FOXC2, they become mesenchymal, which is evident here, and they also develop metastasis to various organs, including bone, liver, lung, brain. But these are all metastases developing from the mammary fat pad to these organs, not these cells are not injected in tail vein. So that's suggesting that, this epithelial mesenchymal program probably a key player in metastasis. Again, Betty Hay and there are several others have demonstrated or suggested that EMT could be a key player in metastasis. So here, just to give a brief introduction about EMT program, this EMT program plays a central role in the formation of mesoderm and the mesodermal organ formation during embryo development. This process also known to be reversal. In other words, the mesoderm transdifferentiates through the reverse process called a mesenchymal to epithelial transition, whereby these cells become epithelial tissue again, such as kidney and ovary during embryo development. It also plays a role in wound healing as well as organ fibrosis. So here is an expression of slug at the wound site, which is shown here in this slide. So these transcription factor, again, the one capable of regulating EMT, tend to be expressed in the mesoderm, which is shown here. This is twist, this is goosecoid, snail 1, zeb 2, zeb 1. They tend to express in the mesoderm or mesodermal tissues during embryo development. So the next part, we want to understand what is the role of EMT in addition to inducing migration and invasion, and we found that it also can impart stem cell properties. I'll show you the data for that. So if you think about metastasis, where most of the time you have a primary tumor and the metastasis, they both histopathologically appear similar. In other words, a pathologist could look at a tumor in lung and predict whether that tumor would have come from breast or prostate or spleen or any organ based on the histopathology they create at the distant site. But we also know that they do undergo this EMT program before they develop metastasis. So therefore, I kind of hypothesized that cancer cells leaving the primary site, they undergo EMT, they become migratory and invasive, which we know, but in addition, they also become stem cells so that they can survive in the circulation, and they can also manage to develop an organ similar to where they come from. So this is again published in 2008. This finding really made a huge impact in the field. And for example, this one shows, this paper has been cited more than 7,500 times, showing that this finding is not only, even though we made this finding in breast cancer, this is now, people have found this similar observation in various other carcinomas beyond breast cancer. So Wenjun Guo, who was also a postdoc in the Weinberg lab, he was trying to understand this role of this EMT program in making normal stem cells. So here, this slide shows normal mammary gland development. The top row shows the cartoon where you could see a duct in the mouse mammary gland growing into the fat pad during puberty. And in a virgin gland, you have the entire duct is filled. And then during lactation, they produce a lot of milk, and then that gets secreted through the nipple. Now that's a cartoon, and this is a real picture in the bottom, which is you could see one of the techniques which many groups developed, including Jeff Rosen here at Baylor, that you can take a mammary gland like this, 3-week-old, and you can chop or remove the ductal tree. When you remove that, now there is no more ductal tree to grow into the fat pad. As a result, this fat pad is rich with growth factors, but they cannot grow a duct. But if you introduce the stem cells, if they have the capacity to develop a duct, they can create a duct. So what Wenjun Guo did, he took differentiated epithelial cells, he introduced slug and SOX9, make them mesenchymal-like, and then he put them into this mammary fat pad assay, which I was mentioning earlier, that you could see even with 100 cells, he was able to recreate the entire ductal tree, but the control cells, even with the 10,000 cells, they did not develop any duct. And here, these cells were able to create K14 and K8 positive cells in the dark, suggesting that through EMT, you could convert even a differentiated epithelial cell to become less differentiated or undifferentiated stem-like cells. So can we target this EMT program or a stem cell program and prevent metastasis? So we know that the cancer stem cells are seen at a clinic at various levels. For example, a clinician could at the time of diagnosis could encounter a cancer stem cell in a tumor, or the cancer stem cells are also related with the recurrence, which I will show you in a few slides. Also, lack of failure is also associated with the cancer stem cells, and I have been showing data that metastasis is also linked with stem cells. So which is shown here again in this cartoon. The stem cells are involved in tumor initiation, they are involved in innate resistance, they are involved in recurrence, but also in metastasis. So if you think about a tumor, again, you have both differentiated cancer cells, which is shown in pink, and cancer stem cells, which is shown in yellow. Now, if you have a conventional treatment, so they will eliminate most of the differentiated cancer cells. Your stem cells will be left out. Now, they can proliferate and differentiate and give rise to a metastasis or a tumor relapse at a later time. Same way, if you target cancer stem cells, the differentiated cells can undergo de-differentiation. If you remember the EMT program, the EMT program can induce de-differentiation or stem cell properties to differentiated cancer cells. As a result, the tumor can relapse or can develop metastasis. The ideal strategy is to combine both, and that's what many groups are doing, including ourselves here at MD Anderson. So how do we target cancer stem cells? So we know that the EMT program provide stem cell attributes to cancer cells. So can we target this EMT program and target cancer stem cells? But if you look at an EMT program, it's a quite complex program. So the EMT can be induced by various factors secreted by cancer-associated fibroblast, tumor-associated macrophages, MDSCs, regulatory T cells, and CD4 T cells. Now, beyond that, if you look within a cancer cell displaying this EMT program, that EMT program could have turned on by many different signaling pathways intracellularly. So it's quite a complex process. How do we target the EMT program to inhibit stem cells and prevent metastasis or treat metastasis? So Brett Hollier, when he was a postdoc in my lab, he found that if you look at a FOXY2, which is the transcription factor I showed earlier, they are predominantly expressed in adipose tissue, but not in any other tissues in adult. On the other hand, if you take this epithelial cell, which I have been talking about earlier, if you overexpress snail, twist, goosecoid, or expose them to TGF-beta1, now they all downregulate this gene called E-carragrin. They also upregulate vimentin, which is clear here. But the most striking finding was that they also, for some reason, upregulate FOXY2. So while we know FOXY2 can induce EMT on its own, but FOXY2 also get induced when these cells are forced to undergo EMT by expressing other transcription factors. So he also found that if you isolate cancer stem cells, they tend to express higher level of FOXY2. Or if you grow cells in suspension, which is predominantly or tend to enrich stem cells in this assay, they tend to also express higher level of FOXY2 compared to the same cells grown in 2D monolayer culture, suggesting that the FOXY2 tend to be high when you force the cells to undergo EMT or if you enrich cancer stem cells by other method. Now, can it be a target for EMT program? But the problem is, Brett did many experiments to test the role of FOXY2 by using an shRNA. He was able to show that FOXY2 is critical for the mesenchymal and the stem cell properties of this EMT program induced by various factors. But shRNA cannot be used clinically, so we need to have an alternative strategy. So Steve Worden, when he was a postdoc in my lab, he was looking for potential kinase which could regulate FOXY2 expression. By this, if you can identify a kinase which stabilizes FOXY2, and if you can inhibit that kinase, if you can destabilize FOXY2, can you inhibit the metastasis and EMT program? That's what he looked for, and he found that FOXY2 tend to have a phosphorylation site for a MAP kinase. It's called P38 MAP kinase. The most interesting thing is if you look in these various cell models, and the cell model which don't express FOXY2, they also don't have an active form of P38, even though they do have a total P38. And that's true even in a human mammary epithelial cells. If they don't express FOXY2, they don't have phospho P38, but they do have total P38, suggesting that whenever you have active form of P38, you also have FOXY2. So they go hand in hand. So if this is the case, if you inhibit P38, what happened to FOXY2 level? That's what Steve asked, and he found that when he inhibited P38 using a small molecule inhibitor in various model system, he found that the FOXY2 protein level reduced significantly compared to their respective control. And one could argue that this inhibitor may be non-specific, so he went ahead and created an shRNA against P38 MAP kinase, and he expressed those shRNA. He found that even the shRNA was able to reduce the FOXY2 protein, suggesting that it is P38 MAP kinase which regulates FOXY2 protein, phosphorylates the protein, and somehow stabilizes the protein. So he also exposed these cells to the P38 inhibitor in an assay called SPEAR assay. In this case, you subject the cells in suspension, and if they have the capacity, if they have a stem cell potential, they tend to form these spheres, which you can see as a colony. But if they don't have a stem cell potential, they don't form any of those spheres. If you expose them to P38 inhibitor, and you can see the spheres which are formed on the left now is gone on the right side following exposure, which is quantified on the graph on the right, suggesting that P38 pathway not only get rid of FOXY2, but also inhibit the stem cell program. So this is the acid test. So now he took the 41 cells which I showed earlier in the where we identified twist and FOXY2. He introduced them to the mouse mammary fat pad and measured their growth over a period of time. He also treated these mice with a P38 inhibitor. What he noticed is there was no difference between vehicle-treated and P38 inhibitor-treated group with respect to their primary tumor formation. The striking observation was that in the vehicle-treated group tend to have a metastasis in the lung. Again, these are going from the mammary fat pad to the lung. And these cells are luciferase labeled, so you can look at the luminescence by looking at these lung. And you could see almost in every mouse he treated with the vehicle, they developed metastasis. But the P38 inhibitor significantly reduced metastasis in these various models. In these various mouse. So, this is from my lab finding, but it's always comforting when you find similar finding from another group. And here is a paper came out in Cancer Cell. In this case, they are testing L.A. Lilly's P38 inhibitor, and they found again in their model that their inhibitor did not inhibit, this is a leukemia, didn't inhibit the leukemic growth in their model. And they were developing a drug called brinapent that also did not inhibit the tumor growth or leukemia formation. But when they did a combination, they were able to inhibit the tumor growth. And this is another study in liver cancer. And you could see the control mice do have a lot of liver tumor. And if they use the Pfizer's P38 inhibitor, it had no effect. They were testing with the sorafenib, that also had no effect. But when they combined P38 inhibitor with the sorafenib, or skepinone, the drug which they are developing, with sorafenib, they were able to see a striking effect, which is shown in the graph on the right, suggesting that this inhibitor does work in combination with other agents, which is what we are doing right now in the lab. So just to complete how the FOXC2 protein works, so we asked, does FOXC2 regulate the EMT program directly? And what we found is that FOXC2 protein also turn on another transcription factor called ZEB1, which is a very well characterized transcription factor known to regulate EMT. And whenever you see FOXC2 protein, they all tend to express ZEB1 as well. What Steve found that when he exposed cells to either express shRNA to FOXC2 or expose them to P38 inhibitor, what he found is that the ZEB1 protein disappeared in various models. It's always a good idea to do in more than one cell line, because otherwise it could be cell line specific phenomena. So overall, this suggested to us that P38 regulate FOXC2 protein, which in turn regulates ZEB1, and that together they regulate this EMT and stem cell program. So, but again, one could argue that it's possible that there may be something else is going on. In other words, the P38 inhibitor may directly inhibit ZEB1, and that is why you are seeing this effect. So to test that, Petra in the lab, she took the cell model which overexpressed snail. They do express FOXC2 at very high level. Here's a snail overexpression to make sure that they do express snail. And they also turn on endogenous ZEB1 because ZEB1 is not expressed otherwise. And they lose E-cadherin and they gain vimentin, fibronectin, etc. Now, if you take this model and you knock down FOXC2 using shRNA, of course, the FOXC2 is gone. And the snail is still there because the snail is overexpressed exogenously. But the ZEB1 is gone, suggesting that FOXC2 regulates ZEB1 downstream of snail. And when you don't have a ZEB1, even E-cadherin is back. So suggesting that FOXC2 regulates ZEB1 and then mediates many of these EMT and stem cell related work. So now recently we have been talking about a concept called epithelial mesenchymal plasticity rather than epithelial mesenchymal transition. So I will explain to you what it is, then I will kind of show its relevance to what we are working on. So this is a study by Jing Yang. Sorry for this, the wrong reference. So what Jing found is that she used a skin cancer model in that she overexpressed an inducible twist, which can be induced by dogs. So when she exposed these mice with the DMBA, the TPA, which creates a skin cancer, now once she made the tumor, then she administered dogs either orally or topically. So what it does is that the oral dogs will give dogs in the systemic, in the entire body. As a result, the twist one, which is induced in response to this dogs, will remain on all over the body. But if you administer dogs topically, then you are turning on the gene only at the site where you apply, and then once the tumor cell moves away from there, they don't see the dogs anymore. Therefore, they will lose a twist expression. The idea is if you keep them continuously on a twist one overexpression, then the cell will not be able to come back from a mesenchymal to epithelial. The topical dogs will allow the tumor to turn on twist, but then it will also help them to revert back. So when she tested under these assays, their ability to develop metastasis, what she found that oral dogs was as very similar to the control mice where there is no twist expression. But the topical dogs expression significantly increased metastasis, suggesting that the tumor cell need to undergo EMT, then they need to revert back to epithelial in order to develop metastasis. So now, this is a study by Jeff Rosen and Jenny Chang and team, and they were treating human patients with various treatments, and what they found is that the tumor cells which left out after the treatment tend to express both vimentin as well as cytokeratin. So in other words, all the yellow cells which are double positive. So they tend to express both the markers. In other words, they do the carcinoma cells, they gain mesenchymal marker, but they don't become completely mesenchymal. So you need an EMT program, but you don't have to go all the way to the mesenchymal, rather halfway through. So this is Max Vicha. He has been talking about, the cancer stem cells where they are something like a mesenchymal-like. They tend to be vimentin positive and EpCAM negative. On the other hand, there's an epithelial-like stem cells. They tend to have ALDH1 positive, but they still express E-character. So in other words, you have a stem cell population which can have, which could have undergone a complete mesenchymal transition, or they could have been midway, but still they display mesenchymal and stem cell properties. So here this study is done in collaboration with Kunal Roy. So we took human mammary epithelial cells, we exposed them to TGF-beta1. So what you see here is that these cells, these are predominantly epithelial, and these are either mesenchymal or the ones which are intermediate. And if you look at a keratin 5, which is expressed in every cell, suggesting that they are still, they are all epithelial in nature. But if you look at a claudin 7, they tend to be mostly in an epithelial cluster. Also the S100A14 is also in the epithelial cluster. On the other hand, if you look at a fibronectin, or this gene called TPM1, they tend to be in the mesenchymal and the hybrid cluster, suggesting that, so what we need to do now is to go deep into the single cell sequencing and then characterize them, the presence of the hybrid cells or the cells which express both the markers. So in collaboration with the HERB11, we have been asking that the activation of this hybrid EM cells or the cells with the intermediate phenotype. So it makes sense, the epithelial cells, they undergo this partial mesenchymal transition, or they just acquire mesenchymal program so that they can migrate, they become stem cells, but once they reach the distant site, they can shed off that mesenchymal property they inherited, and then they can lose, they can become an epithelial. They can also become mesenchymal in some instance, such as fibrosis or in other scenario. So we have been trying to understand the role of EMT versus EMP in a chemoresistance and metastasis. A lot of studies ongoing right now in the lab. A number of postdocs are testing that. So the next, in the next few minutes, I'll show how to identify cancer cells with the mesenchymal and stem cell properties, because we need to know the cells with both the properties to diagnose them. Then it'll be easier to treat these patients. But if you think about a tumor, most of the time when we study tumor biology, we isolate human cells or tumor cells. We just study the tumor cells by itself. While that would give us some information about the tumor cell intrinsic phenomenon, it would never tell us what you see in a tumor in a patient where you have natural killer cells. You have macrophages, you have MDSCs, platelets, CD8 T cells, B cells, CAPs, adipocytes, etc., etc. So the ideal scenario would be using a right model to study that. But, it's difficult to comprehensively look at all the population and then identify the carcinoma cells which is displaying the mesenchymal properties or stem cell properties. This is evident from one of our study. So Joe Tabe, when he was a postdoc in my lab, collaboration with Jason, when he was in Jeff Rosen's group, they took breast epithelial cells, which are undergone an EMT through this various method. They took these cells, and in collaboration with a number of folks listed here, we performed a microarray. We generated an EMT signature. So when we went back, used the signature, and went back to human patient material, a tumor, and we asked, can we find tumors with enrichment of the signature? We could not. We could not use the signature to identify a tumor which could potentially develop metastasis or chemoresistance or tumor relapse. But what they were able to do is to predict that a tumor type called Claudin-Low, which is predominantly mesenchymal, they were able to identify that tumor subtype easily using the signature, suggesting that if the entire tumor is made of mesenchymal-like cells, you can identify them, but there's a subpopulation in a tumor display this phenotype, you cannot. So this is a nice paper which kind of shows using a single cell omics, they were able to identify cancer stem cells, differentiated cells, immune cells, etc. So what we are trying to do something similar. So one of the approach here we are doing, this is in collaboration with Dr. Vinstuba's group, who's my chairman of a department, also a division head, that when they do immunostaining, multicolor immunofluorescence for cytokeratin, which labels tumor cells, PD-L1, PD-1, CD3, CD8, of course DAPI for identifying the cells, and CD68, and you can see the tumors tend to have varying spectra. And again, this is also the tissues were originally generated by Stacy Molder, who's conducting an innovative clinical trial called Artemis. So what we need going forward is that we need to use this kind of single cell omics approach plus multi-color imaging by combining the two, because the single cell RNA-seq will tell us presence of such a cell, but it won't tell us exactly where that cell is present. Is it present in the middle of the tumor, periphery of the tumor, sitting right next to CD8 T cells, or sitting next to an MDSC? All those information cannot be gathered at this point using this, but this will give us, however, still it will give us quite a lot of information about the cell type. This image or this kind of analysis will give us a little more information about the spatial orientation of the cells. So by combining these various methods, we would be able to do it. So what we are doing right now is using this immune panel, EMT panel, doing an immunofluorescence. Simultaneously, we are also characterizing these tumors at a single cell level. So we would gain quite a lot of knowledge about these cells. But at this point, we don't have a best way to diagnose patients with the EMT and stem cell properties because we don't have technology available, or we don't have a developed methodology available. So just want to end my presentation with one of the resources which one of my postdoc developed. His name is Suhas Vasekar. He developed this tool called EMTome, which is quite a nice tool, and we are trying to get it published, but you are all welcome to try. So what he has done, I'll just give you a small kind of analysis what one could do. So this kind of list shows the number of papers which are getting published in the area of EMT alone. And this graph, which you will see in the EMTome website, shows the number of papers published in different cancer type on each gene. So each one is a gene. So one of the highly studied gene in various cancer is twist. You can see that this is shown here. So what he did, he's able to generate with the help of various people in the lab. He generated a EMT core signature, or he collected various signatures derived by various groups. And he also generated a list of genes known to regulate EMT. And he created kind of EMT gene interactome, various analysis. I think I would recommend you to take a look. And then he applied that into pan-cancer analysis, applying clinical mutation, CNV, methylation, microRNA, mRNA, etc. So by combining all this, for example, here is a nice one of the output which this program gives. So if you ask what immune cell type positively or negatively correlates with ZEB1, so here you can see ZEB1 expression and TH17 strikingly inversely correlates with the ZEB1. So you can do quite a lot. So I would strongly recommend all of you to take a look. And in summary, so what I have shown is that the EMT program plays a central role in regulating metastasis. The EMT program induces stem cell properties on differentiated cancer cells. And the targeting EMT can prevent metastasis. And we are working on the chemoresistance part. The P38 signaling is active in cancer stem cells, and inhibition of P38 can also inhibit stem cell properties and metastasis. Finally, the EMT program, it also modulates immune cells, and the immune cells imparts EMT program. So therefore, it's extremely important, because I know that a lot of students are also in this call. So I would strongly recommend you all think twice before you design your experiment if you're trying to study metastasis, to make sure that what kind of model system you are using and what kind of question you are asking. That would really help in addressing or answering the question. For example, if you use a cell line and if you inject them through tail vein, you will not be able to understand the metastatic niche at the primary site, right? But you can look at it at the distant site. And you cannot study invasion. And then most importantly, we cannot study immune cells when you put a human cells in mouse. So I would recommend you take a look at this paper. So what we are doing in the lab right now is, we do know if the tumor tend to be differentiated or epithelial-like, they are sensitive to immunotherapy. We know in most cases, except in breast and a few other disease, they tend to be sensitive to chemotherapy. They are not metastasis prone. They don't. The tumor relapse is low, and the patient survival is really high. But if you tend to see more mesenchymal or stem-like cells within a tumor, then they are not sensitive to immunotherapy or chemotherapy, and they are metastasis prone, tumor relapse prone, and most importantly, the patient survival is extremely low. So these are moving targets, right? They are constantly changing. When you treat a patient, now the tumor cells probably like this, and over a period of treatment, they switch to more mesenchymal. So this is a moving target. How do we target this moving target? It's a challenge, but that's something we are exploring. We need to understand the interplay between cancer cells and its tumor microenvironment. Again, it's extremely important to see how the tumor cell is talking to immune cells and the rest of the tumor microenvironment, and how the tumor microenvironment is talking back to the tumor cells. Here, cancer cells capable of undergoing EMT are, again, that's changing the microenvironment, and it is also changing itself. So it's extremely important for us to understand both. This is all this study, what I'm talking about is not possible without the support and blessings and collaboration from colleagues and mentors along the way. So I talked about some of the work which is going on in the lab, but not all. So we are working with Massimo on circulating tumor cells. Stacy Molder, who was an instrumental in this clinical trial called Artemis, which is really feeding us quite a lot of information about the biology, what we are studying. So when we study something, and when you can understand what you are studying has a clinical relevance, it makes you feel that your time is well spent. Herbie Levin, who's a physicist, and then with whom we are collaborating, and he's doing a lot of modeling and understand this hybrid EMT, and we are also trying to examine or experiment the role of dormancy in this process. And Dr. Vistuba, who's been an amazing mentor, provides various support for us to, not only a very good environment, but also providing support to constantly scientific advice, various tools to explore these EMT and stem cell concepts. Jeff Rosen has been a mentor here when I came to Houston. Is my faculty mentor, and he has been instrumental in help shaping various studies in the lab. And of course, Bob Weinberg, who's an amazing mentor. Most of the time, when you join a lab, I tell my people in my lab that it'd be great if they could come up with their own project so that they can take that with them. And that's what Bob Weinberg allowed me to develop these various ideas and take that with me and to develop that further in my group. And thanks to Bob. Again, this is I collaborated with various folks at MD Anderson, Baylor College of Medicine, IBT, also Jeffrey Chang from UT Health Science Center. And I collaborated with the Scripps to do a small molecule screen. We identified small molecules capable of inhibiting the CMT program, but I didn't get a chance to talk about that. And this is my group in 2019. You can find a lot of information by visiting money.us, where that is connected to the MD Anderson Money Lab website. And of course, it's the most important slide of my entire presentation is this. It's tough to get funding, but at the same time, without funding, we cannot do the work. And special thanks to various organizations. I want to thank Bose Foundation. I visited them, I spoke to them, and they really kind of impressed, and they gave us support, and which we are able to use to do a lot of these innovative studies. I thank you all, and it's a new reality, virtual presentation and sitting in your comfortable desk listening to a talk given from somewhere else. And hope you all enjoyed. Take care, and we'll open for a discussion. And I think this is Q&A.
Thank you. Thank you, Dr. Mani. Thank you for this very exciting, wonderful lecture. It's been a pleasure to listen to it. I have some questions from the audience. I will read them to you without the name of the person, since this will be then placed online. The first question is, if you have decreased epithelial markers, but not as much change in mesenchymal markers, how would you consider that setting? Is that partial EMT? What might be the biological relevance of that?
It's a good one. It's a good question. So the question is, if you lose epithelial properties, but not mesenchymal properties, right? So what is the biological implications of that for patients, for the treatment, right? That's if I understood correct. So if you are losing epithelial characteristics, which means the differentiated cells are getting killed, and if you are gaining more mesenchymal properties, which is what, more of an EMT program, as you all know, most of the chemotherapy does induce inflammation within the primary tumor, and that could, while killing the predominantly differentiated epithelial cell, you tend to enrich those mesenchymal cells. So, which is bad, clinically speaking. I hope that I answered that question. So in other words, losing an epithelial and gaining mesenchymal is bad.
Yes, thank you. There is another question. First it says, great talk. And then the question is there a formal proof that p38 induces spontaneous metastasis via FOXC2? Also, does active p38 correlate with FOXC2 in human metastasis? And is active p38 prognostic for metastatic tumors in patients?
Thank you. It's a great question. So that's something which we have been battling with. So the problem with the p38 is there are 4 isoforms. And at the moment, we have the antibody which recognizes phospho-p38, also recognizes other isoform. As a result, we have been struggling to have a right biomarker to identify the p38 MAPK14 isoform, the active form of that. So one of the things we are doing, because now we have access to multiplex, so we're trying to do co-staining of phospho-p38 antibody FOXC2 as well as MAPK14, which is a total p38. And once we have that, we will be able to predict better. So this is one of the, the patent lawyer, when you file a patent, that's one of the major concerns they have. The roll-up, the first 2 part is that, could you please repeat that first 2 parts?
The first 2 parts, is there a formal proof that p38 induces spontaneous metastasis via FOXC2?
Yes, so that we have done additional experiments, which I didn't have a chance to show, that we overexpressed FOXC2, which cannot be phosphorylated by FOXC2, or we also overexpressed FOXC2, which is phosphomimetic, which mimics the phosphorylation. And then we conducted an experiment and exposed them to p38 inhibitor, the phosphomimetic one didn't care about p38 inhibitor. The non-phosphorylatable mutant did not develop metastasis. So that argues that the p38 pathway regulates metastasis through FOXC2. The second part of that question? Sorry.
Was it, does active p38 correlate with FOXC2 in human metastasis?
That's the one problem. Again, it's a lot. We since the lack of proper tools, we are not able to do that. But we are in the process of doing now by combining p38 MAPK14, phospho-p38, and FOXC2. We are doing a co-staining and looking for presence of such cells. Thank you.
Thank you very much. Thank you. And
Mani, great
yeah,
talk. I still have a question. So wonderful talk. I enjoyed. Mani, it's something known about the role of neurons who surround or are infiltrating the tumor and the EMT.
Thank you. It's a very good point. There is a number of people are trying to understand the role of neurons and its involvement in inducing this epithelial mesenchymal transition. But until now, we're not successful. There are only a couple of studies, a few studies showing that neurons could potentially stimulate this program. The issue is how the neuron is changing. So we know if you take a fibroblast, if they are growing right next to tumor, over a period of time, they become a carcinoma-associated fibroblast. They become converted into something which supports tumor, which is also true in case of an MDSC. They do start supporting tumor. The macrophage, again, one form of macrophage inhibits tumor, another one promotes tumor. So they all get converted to something which supports tumor over a period of time. In other words, tumor educates those cells. So I heard it from someone that people are studying how the tumor can educate even neuron to become tumor supportive. But to my knowledge, I have not seen any evidence, and we are not working on that. Thank you.
Thank you very much. Thank you. Thank you.
Thank you, everyone. I think this concludes today's exciting lecture. We'll see you back next week with more presentation as part of Enjoy Science seminar. And we would like to thank you, Alison and Liz, for their wonderful technical support. We wish everyone to have a good weekend and stay safe.
Thank you. Thank you so much. Thank you all the participants. You all stay safe. And thank you, George, and take care. Yeah, thanks.
Thank
Bye.
you.